know the most important constraints to be addressed in a particular field, and then to be able to gauge changes or monitor improvements with time as we change management practices in the field, for example. Sometimes we might want to have an overall index of overall health or functioning of a field. And then again, we think we would like to monitor over the long term to see how changes occur over the years and decades, much like we do with standard soil fertility testing. I do want to remind us all that we have been measuring aspects of soil health on and off for quite a long time. We just maybe didn't call it soil health at the time. So in my research group, for example, we've done earthworm counts for decades. We've measured penetration resistance or soil physical constraints to rooting with a penetrometer. We've measured water infiltration rate. How fast does the water go into the soil? All of those are components of soil health. Primarily here the physical infiltration and penetrometer, but the earthworms is one of the biological measures. So let's not forget that we do actually have quite a history of measurements that contribute to our assessment of soil health. But what we're really going to focus on today is some of the newer tests on soil health. So in Indiana, we had the CCSI Conservation Cropping Systems Initiative had a conservation innovation grant from NRCS that began in the fall of 2012. And the motivation for this, for us writing this grant, was we were getting lots of questions about some of the new soil biology and soil health tests from farmers and our conservation partners and others. So this was in 2010, 2011, 2012, we were getting these questions. And these questions were things like, how useful would these tests be? Could they help us distinguish our different management systems? Could they monitor changes over time? Would they be worth the money because they're pretty expensive compared to a standard soil fertility test? So those were the questions that motivated our project. And the commercial soil health tests that we were looking at were primarily 3 of them. For biological tests, the phospholipid fatty acid or PLFA test is one of those that looks at the fatty acids in cell membranes. So it's a biochemical marker for living cells and a little bit of a fingerprint, although not a perfect fingerprint. The Cornell test, which is now referred to as the CASH, Comprehensive Assessment of Soil Health, really does physical, chemical, and biological measures. And then the Haney Soil Health tool does chemical and biological. So those were the 3 that we looked at. There are others available as well and new ones that are being developed with time. Today, what I'm going to do in my very brief time is give you just a few examples of some of the commercial soil health test results that we got in Indiana for PLFA, Cornell, and Haney. Obviously, in 20 minutes, I can't give you the complete story. I want to talk a little bit about challenges of using and interpreting those tests, and then recommendations based on our experience and our results. So our Indiana Conservation Cropping Systems Initiative, many of you are aware of that, but for this particular project, we had 17 sites. I'm assuming that you can see my mouse when I move it, right, Paul? You're going to nod your head and let me know that you can see my mouse if I am moving it? Yes, okay.
Yes.
All right, thank you. We had 17 sites across Indiana. 12 of them were farmer cooperator sites. 3 of them were Purdue Ag Centers, and then we had 2 SWCD sites. We had a variety of treatments, but most of the sites, their main treatment was cover crop versus no cover crop strips, replicated 3 or 4 times on a long-term no-till site, and that's for better or worse, which I'll talk about here in a minute. Because we had multiple goals for this project, one of which was the research, one of which was education of farmers. We wanted to have farmers as our cooperators here who could be part of our teaching cadre, who were already quite experienced with soil health management systems, including no-till and cover crops, not on this particular field. So we chose farmers who already were into no-till and had a field that had not yet had covers on it. We did succeed in getting a few conventional neighbors for a few of those cooperators, but not as many as we had hoped. The soil health sampling that we did, this project started in the fall of 2012. We collected in the spring of 2013 our first set of samples, which was baseline samples for most of the fields. They had not yet had cover crop, and then they planted their first cover crop for this project in the fall of 2013. The timing for our sampling was generally in early June, V3 to V6 stage of corn. It did vary somewhat among some of the sites. We did sample to our full depth that we usually sample in Indiana for soil fertility, which was 0 to 8 inches. And in each plot, we basically collected about 30 soil probes, mixed them together, homogenized them, and then shipped them on ice packs to the commercial laboratories as required for good samples. So again, sites were placed throughout the state of Indiana. We basically had the 4 quadrants of the state, and we had 3 or 4 sites in each of those. Okay, so getting to just a few of examples of the data. We'll look first at a couple of results from PLFA, phospholipid fatty acid, and that test, for those of you not familiar with it, will break the organisms by their fatty acid profile into different general classifications. And then there's a bunch of them that they can't identify. So this was the classification of total bacteria and total fungi. So the graphs I'm going to be showing you will have the particular parameter that's measured. And I will be showing you a couple of the sites that we had, usually farmer sites. So if we take a look at total bacteria at the DeSutter site in northwestern Indiana, the legend or the axis on these graphs, if you look at this, we have no-till plus cover crop and no-till. So that was at the DeSutter cooperator field, those 2 bars. And then in this particular case, we did have a neighboring field, and that was the best neighboring field that we could have because it was immediately adjacent to his field. So, and then the standard statistical notation here, you could see that our no-till alone and no-till plus cover crop were not significantly different in total bacteria. The no-till field was significantly higher in bacteria than the neighboring field. What year is this? The year is 2016. This happens to be the 2016 samples taken in June. The total fungi were not significantly different among any of those 3 treatments. At the Vilwalk site in southwestern Indiana, there were no significant differences. He also had a neighbor who was a little bit further away, but similar soils. No significant differences in total bacteria or in total fungi between the no-till plus cover and the no-till in his field and the neighbor, okay? We did use a more generous significance level of 10% instead of 5% because of the nature of the field data. All right, what about mycorrhizal fungi? The DeSutter site, the no-till with and without cover crop were not different from each other, but were significantly higher than the neighbor. At the Rulon site, he had a field, a no-till field where he had 3 different covers: annual ryegrass, cereal rye, oats radish, and then no cover crop, okay? And in this case, all 3 of the cover crops were significantly higher in this 2016 sampling than was the no-till. All right, that's just a snapshot of a few PLFA results. They were not consistent across, farmers or regions or other things. So let's move to Cornell and Haney tests. And what I'm talking about here is that both of those tests have a couple of the things that they measure are getting at the same function. They're trying to assess the same function. So Cornell has an active carbon measurement and Haney has a water extractable organic carbon measurement. They are different chemical tests. We're not saying they're identical, but both of them are trying to measure this fraction of soil organic matter that's easy for microorganisms to use. So they're trying to get at what is some of that easy food for microbes that would then allow them to release nutrients. Another test that they both do is soil respiration, which is the same test. It's basically measuring potential activity of microbes in the soil, how much carbon dioxide is released in a certain amount of time. the difference between the two tests is Cornell does it for 96 hours or 4 days, and the Haney test goes only for 24 hours, and there's pluses and minuses to that. All right, so let's look at the results from a couple of sites with these similar purpose measurements. So readily available carbon, we have the Cornell up on the top, and we have the Haney on the bottom. Going to our DeSutter site again. The Cornell active carbon measurement showed that the no-till field with and without cover were the same as each other, but were significantly higher than the neighbor. But the Haney water extractable test, which is also trying to get at that easy carbon for microbes, did not show any difference at all among all three of those treatments. If we go to the Rulon treatment, no significant difference among the three covers and no-till alone, whether it was measured as active carbon or as water extractable carbon. At the shooter site, we had a, I have to get rid of all these pictures that are in my way. There we go. At the shooter site, he had strip till plus cover crop versus strip till alone. The active carbon measurement did not show any difference, but the water extractable carbon did. So again, the point here is that both of these measures are trying to get at something fairly similar, but they don't give us the same answers. Let's look at soil respiration, which really is measuring the same thing just over a different time period. So the Cornell test, again at DeSutter, the no-till with and without cover were similar to each other, but higher than the neighbor. The Haney test over a shorter time period also showed the same trend. No-till with and without cover was higher than the neighbor. If we go to the Rulon site, the Cornell test did not see any significant difference among all those treatments, whereas the Haney test showed that the oats radish was significantly lower than the annual ryegrass and cereal rye, and significantly lower than no-till alone without any cover. At the shooter site, the Cornell test did not show any difference. The Haney test did show a difference. So again, even the same test does not necessarily give us the same results and the same implication. Both of the Cornell and the Haney tests do some kind of an overall quality score. The Cornell test gives you this stoplight type of thing. We have our 4 physical measures, our 4 biological measures, and our 4 chemical measures. This column here, which is not highlighted, gives you the actual measured value for each of those parameters. And then the rating is a rating between 0 and 100, and you got a stoplight kind of thing here. If it's green, it's really good. If it's red, it's really bad, and the intermediate colors are in between. So the first thing you can notice is these green 100% rating. They're all the chemical things. They're all the things that we farmers and all of us have been managing really well for decades, right? We know how to handle soil fertility, and it reflects right here. We're doing a great job on soil fertility. What we're not doing quite so good of a job on is some of the biology and the physics. So soil respiration was very low, and aggregate stability was very low. So it gives you an idea of what are some of the things that you might want to work on a particular soil. It also gives you a weighted average of all of those 12 scores, and in this case, the overall score was 66, which they said was excellent. But I'll tell you that in my soil physics class, 66 would not be considered excellent. But anyway, this gives you an overall score, which could be potentially a value as an overall ranking. But again, what I really want to know is what are these things that I need to work on, right? So the individual measurements are really much more useful than the overall score. All right, so let's look at that Cornell overall quality score and the Haney overall quality score. I didn't show you the calculation on that, but basically they calculate with primarily the respiration measurement and the water extractable carbon measurement. Okay, so again, at the DeSutter farm, the Cornell score showed that the no-till with and without cover crops was significantly higher than the neighbor. And the Haney score also showed that the no-till with and without covers was significantly higher than the neighbor. So there, at least they were giving us similar kinds of implications. At the Rulon site, neither the Cornell test nor the Haney test showed any significant differences with 3 different covers versus no-till alone. At the Shooter site, the Cornell test did not find any difference between the treatments, whereas the Haney test did. So again, we don't necessarily expect that the quality scores are going to give us the same answer because they're based on different kinds of measurements. But again, it just reemphasizes that a high quality, overall quality with one test is not necessarily going to mean that for other tests. All right, so I'm going to get into some recommendations, but let me just summarize these data and the many data that we have. There were relatively few differences between cover and no cover treatments. However, remember that most of these sites had only 3 years of cover crops that were added to long-term no-till fields. So they were already probably in a pretty good condition with the long-term no-till. We did find more differences between no-till with and without covers compared to tilled conventional neighbors where we had them. One of the frustrating parts was that some of the tests that we expected to show similar results to each other did not. So I'm going to go through a couple challenges and then a couple recommendations. So I might be at 21 minutes instead of 20, but I'm just going to forge ahead.
You're okay. Go as long as you want.
Okay, thanks, Paul. The challenges of soil health tests is, first of all, they're snapshots in time, which, of course, that's the same with soil fertility tests. But if you're thinking about biology, snapshots in time is really important because of the biological organisms and their response to temperature and moisture. Interpretation of soil biological measurements is really challenging because we have no thresholds. If you have a soil fertility test, you know what a phosphorus test of 20 means, right? Do you need to add fertilizer or not? And that's been built on decades and decades of work. We don't know what does it mean to have 2,000 protozoa. Is that enough? Do I need more? And that kind of thing. And variability is really, really important with biology because organisms tend to be patchy in fields for small differences in topography and other such things. So let me go through my recommendations real quick, and then I'll give you the extension publication where you can read more about these. First of all, choose your lab and your tests and stick with it. I realize that because these tests are new, that may not be possible, but the reason I say this is the PLFA tests, we had to change labs for a variety of reasons. And they measure things slightly differently. They use slightly different fatty acids as the markers for different groups of organisms. And the units were not interchangeable. One was in nanograms and one was in nanomoles. And we could not interchange them. So as much as possible, choose your lab and test and stick with them. Pick your spots. And by that, I mean that these are very expensive, right? These are $50 to $110 per sample. You're not going to do grid sampling. So what we would suggest is you choose a few locations in your field. Maybe you choose, high yielding, low yielding, and somewhere in the transition. You mark those points. You pull multiple samples from a 20 or 30 foot diameter area. You combine them. You send them to the lab. And then the next time you go measure that 2 years or 3 years later, you go measure the same points, okay, to see whether you're making progress. Be consistent, and be consistent, I'm going to talk about 2 different parts of that. One is on the timing. This was one of my master's students, Jennifer Woodyard, who measured some of the biology at a couple of sites. So this happens to be PLFA measurements at the DeSutter site. And you can see here the orange line is no-till plus cover crop, and the blue line is no-till alone. There was one time period which happened to be V3 to V6, so we kind of guessed right, I guess, on some of our other sampling. There was that one time period where there was a significant difference between with and without cover crop, probably related to the fact that the organisms were really chowing down on the cover crop and decomposing it, and one of my colleagues has been measuring enzyme activity, and it's really much higher at that point in the cover crop decomposition cycle. So if your goal is to measure what kinds of differences there might be, then you might want to measure at that time period. If your goal is to measure the maximum amount of activity, then it might be actually after harvest. But clearly you can see you don't want to be taking samples 1 year at V3 to V6 and another year after harvest and try to compare them. The other thing about being consistent, which as soon as we put these pictures together, it was very obvious, but be consistent on the cash crop you're sampling in. So this was the Jamie Scott site, which he has these cocktail cover crop mixes, you know, so he's got no-till plus a cover crop mix, no-till plus one cover crop, and no-till alone versus a neighbor. And so we expected to see a lot more activity in the Scott field than we did in the neighbor. The problem is that that particular year he was growing wheat and then going to seed his cover crop cocktail mix, and the samples were taken in July. Well, it's very obvious when you see these pictures. This was sampled in dead wheat stubble. The neighbor was sampled in a growing, thriving soybean crop. So, of course, now in retrospect, we know that the neighbor at that moment had the neighbor in the soybeans had a lot more biological activity, both total fungi and protozoa, than did the Scott farm in the dead wheat stubble. Track changes over time. I don't have time to show you data, but Stacy actually has a paper we're about ready to submit that shows some success in tracking changes over time. It happens that the changes with no-till and with no-till and cover crop were similar over time, but we did see some changes over time. Interpret with caution. This is mainly related to the extremely high variability. And then focus on large management changes. So if you're going to do these kinds of tests, I would recommend that you don't do what we did, which is take a long-term no-till field, impose cover crops for 2 years and expect to see a change. But rather look at maybe somebody who's converting from conventional tillage to no-till plus covers and watch that kind of a change with time. You're more likely to see these soil health differences. So to just list those recommendations again. Pick a few spots, be consistent in your timing and your cash crop that's sampled. Try to look at changes over time and focus on those large management changes. So this is my last slide, and I'm going to leave this up. We have an extension publication which essentially goes into more detail on what I just talked about, which is available for free download at the Purdue Education Store. We also have an article in the Journal of Soil and Water Conservation. If any of you are contemplating doing these kinds of on-farm soil health tests, I really encourage you to read our article. We had a lot of great cooperation, but there's a lot of challenges to doing that kind of a study in a way that's going to give you good data that you can really interpret. And so I would love it if you would learn something from the lessons that we learned. If you're contemplating starting, or if you've already started something like that, maybe there'll be a few tips in there that might help you out.